And then this one should go away. All right. So basically, we're covering all the protein stuff, right? Amino acids, their structures, and the polypeptides, proteins, their higher level structures, from the primary to secondary to the quaternary structures. So we're gonna go over the practice exam questions first, really quickly, just in case you guys have any questions or the confusions or anything. So what would be the answer for the first question? Number one. Yes, it is. It's E, and it's pretty obvious when you look at the protein structure, right? And the reason that I actually, made it as a exam, practice exam question was that I want you to actually go over all the other structures or some important functional groups and their features on the different amino acid side chains. Second one is the tyrosine, basically. And again, you need to know the both 3-letter codes and the 1-letter codes for the amino acids. It's pretty simple. I mean, basically asking quite similar things. What about the question number 3? What would be the right answer? Oh, here's a question. That's a good question. We actually have the structure. We covered it during the lecture. I believe that's protein 1 at the end of the protein 1 lecture, where we talk about the other derivatives of the amino acids that can work as a different stuff, like the neurotransmitter and other things. So you can actually go there and see it, and you can also see that the name itself, it's thyroid hormone, thyroxine, and typically you actually use a quite similar naming for the derivatives of the original amino acids. So that could be another clue. All right, what about the number 3? What is it? D? So it's D. Any confusions on here? Why is it not B? Can anyone explain? So B says pKa's that assure the solubility of every protein. Anyone? Exactly. So it's never gonna be soluble, right? And pKa itself is not about the solubility. It's about the what is it? The tendency to release a proton, right? It's a dissociation constant for acid. So, I mean, typically you get ionized when you release a proton. That means you can be a little bit more soluble, but that doesn't apply to all amino acids, basically. That's why. All right. Oh. I didn't catch the first one. Are you talking about are you talking about B or D? Yes, it is. So for certain amino acids, and they're buried inside of the protein, right, in the core, which are mostly hydrophobic, which is a different environment other than the amino acids that are exposed to the aqueous solution, where there are lots of ions and water molecules are. So that's why that changes the propensity of releasing the proton, right? So that's why. Any other questions? All right. So question number 4. Would it be the right answer? I hear that there was E, so hopefully all of you said E. So it's pretty much everything, right? Number 5, it can be a little bit tricky. What is the right answer? Any questions on this or anything? How is what? How is it B? It's about the planar property of the peptide bond where it is sort of fixed, right? So it's all about when you draw that plane, the region, that means you want to talk about the rigidity or the inflexibility of that region, around the peptide bond. It's not about other stuff actually, which is, I mean, you can talk about the rotation angles and stuff like that, but it's outside of that plane actually, right? So the purpose of highlighting that region is because we want to tell that that's the fixed region where it is formed basically conferred by the pi orbital, where this double bonds between the carbon and oxygen molecule is sort of migrating towards the peptide bonding right here, forms again cloud electron clouds up and top and bottom of that plane. Does that make sense? Questions? Okay. Number 6. I hear B, C, D, but it's D. So disulfide bond, when you look at the two cysteines, they contain the sulfhydryl groups, and then basically they lose the hydrogen atoms there, right? So that's losing the hydrogen, meaning that deprotonated, which is the same as oxidated. It's based on the acid base, definition. E, which is SS bond, is a special form of a peptide bond linking two cysteine residuals. Can anyone answer to this question? I didn't hear the sort of middle part. Can you speak up a little? Exactly. So that is not a peptide bond. It is not. The peptide. What is the peptide bond? The peptide bond is right here. The link between the nitrogen atom and the carbon atom. And they shouldn't be the center carbon or the chiral carbon, which is the alpha carbon. Shouldn't be, All right, so. Number 7. Answers? Oh, there's a question. Can you speak up? So I heard the first part where you're saying peptide bond shouldn't be. No, it is not. So the peptide bond, alpha carbon is right here, which actually has the bonding to the all other functional groups. So it's in the center, chiral center, right? Where it has a carboxyl group and an amino group too. And the peptide bond is formed between the carboxyl group and amino group, not the alpha carbon and amino group. All right. Number 7. Oh, there's another question. Oh, no. C is the right answer. So it's pretty straightforward. So I'm going to just, go ahead. Number 8. It is D. So Pi is basically an isoelectric point, right? So it uses that property. And it basically precipitates protein when the protein gets to the Pi, right? Number 9. It's the C. What would be the last step among here? Oh, here's a question. Why is it not E? Can anyone actually answer to this question? Yes, that's correct. So the first step will be the forming a smaller like domains or even secondary structures where they form it, sort of making the protein core. And protein cores are basically hydrophobic hydrophobic regions, right? So that's why hydrophobic is at play to the forming the core first, and then it'll fold the another regions basically. And I'm not just talking about the protein core; it can be the domain cores too. So multiple small regions within the protein. Yeah. So the hydrophobic effect happens first, and then you try to find. So that's sort of my question, right? I was asking what would be the last step, but what would be the next step after C? So there's an aggregation of the small regions, which are domains. You know, it could be just a secondary structure too, in some cases, or super secondary structure, but mostly domains, which have the sort of independent, like modular structure. What would be the next step among these four? B and Y? What are the subunits? Subunits are basically a component that forms the higher order structure, like like some kind of protein bundles, any kinds of things like alpha keratin, for example. It makes the really kind of big structure, super protein high ordered protein complex. So subunit can be just one protein actually, and be a component for those kind of huge complex. So not necessarily the second step, right? As you can imagine, I mean, you have to have a protein first to build it as a big structure. The last part? Mm-hmm. Yeah. But typically when you talk about subunits, when you call subunits, those are the independent, like functional protein already. That is a sort of a building block for the high order structure, like some kind of scaffolds or some kind of sheet or things like that. So probably the last step would be B, actually. But after it forms the core, hydrophobic cores on the domains, which involves the formation of the low energy and low entropy too. But at that stage, it actually starts with the pretty high entropy. If you remember that landscape for the energy, right? So the first step is again the aggregation of hydrophobic regions. The second step would be the getting the tertiary structure first. But just forming the tertiary structure based on the finding the low energy state, which is the same as low entropy state. And it'll go through the refinement to try to find the native state, which is the most optimal state with the lowest energy, right? So it forms the one protein structure. And after that, it'll combine with the other subunits. If I have to, that would be C first, and then A and E at the same time, and then D, and then B. And I mean, it's just like sort of general cases, right? Sometimes it happens in the sort of different ways, but absolutely C is the first one, and B is the last one. It typically starts with the highest entropy, as you know, and then it gets lower and lower. But of course, you sometimes gets the dip, right? There's a local like minima, basically, where there's an energy dip there, but it's not the native state. So you have to actually have some kind of input by the chaperones or some other biochemical reactions involved there to get out of that minima, local minima, and then have to get to the actual native state. All right, what about number 10? Answer? B. Is it clear? Number 11. Tertiary structure. So it's only the difference is the whether it is quaternary. It makes the quaternary structure or not, right? Whether this one blob of protein works as a subunit. right? For the hemoglobin or not. Number 12, B. 13, it's D. How come? Exactly, exactly. So you have the in that region, you definitely want to have the hydrophobic region, right, which is non-polar. So the D is the only non-polar protein, amino acid there. 14. Yeah, just have to memorize this. Number 15. Proline. Again, as long as you understand and know the structure of the amino acids, you can answer to this question. 16. It's E. And how come? Can anyone explain why E is the right answer? Or is it too easy? It's acid, exactly. That's why. I mean, it at least they have some charged side chain, right? So it could have been anything like charged amino acids, basically. Number 17. It's E. I think I heard D question. Numbers? It's E. Mm-hmm. I'm sorry? So, well, I mean, it could be the just definition of being acid. It's not a measurement. When you talk about, it's something, some substance is at pH 7, that doesn't mean that that substance itself has the pH value as 7. It means that that substance has placed in or at pH 7. So it's a physiological condition. Now it can behave either acidic way or the basic way, depending on its pKa value. But if it is acidic amino acid, meaning that it's prone to lose proton, it'll lose it pretty easily at the physiological pH range. And of course, I mean, even if the environment goes a little lower in terms of the pH, it can still have a capacity to release it. So it's a relative concept rather than it actually defines the itself, the substance itself for its pH value. Would that make sense? Exactly. So, I mean, you can't measure pH of, what is it? Glutamic acid here, E. You can't do that because the pH is the state, right? So if there's some kind of solution, you can measure pH. But if you just put one substance in there and measure the pH, that's impossible. You can measure pKa, and you use the Henderson-Hasselbalch equation basically to estimate the what kind of a pH would be the point where it releases a lot of protons basically, the inflection point for the titration curve and things like that. But the pH value will only define its environment, the solution. So the more acidic ones will release protons at the certain pH, like physiological condition here, as opposed to basic ones will accept protons. Hope it makes sense. If not, then we can talk more basically, because it's a sort of a principle kind of the question where the maybe requires more explanation then if you don't actually get this, and it's really important to understand this. So, all right. So, did we talk about 17? Okay. So basically the same reasoning applies to this, as 16, number 16 too, right? Yeah, as it says, it's E, which is not, which means that all of the others can be ionized. And how come? What does being ionized mean? You got it? Anything to add? I saw hand raised somewhere here. So same for tyrosine? Yes. Yes, but it has a little less capacity to be ionized because of the phenol ring. But exactly. So that's gonna be anything ionizing, meaning that you either lose the proton or accept the proton, or the losing the hydroxyl group, things like that, right? So 7 total is it? So there are, yes, 7 in total. So maybe 8, because that includes the all acidic and the basic amino acids, which are 5, right? Because they are charged, of course, and including the something that can happen on its side chains, like serine and threonine, basically. Those two have hydroxyl groups that can release the proton there. Tyrosines will do a little bit, but again, not at the capacity as other amino acids can do. So you can include it in certain cases, but it's not going to do that too much. Yep. 16, actually, the gentleman sitting there explained it really well. So the cysteine contains a sulfhydryl group, which can release the proton there from sulfhydryl group. So it can form the disulfide bonds. So that's why. Okay, number 18. Hopefully it's relatively easy. And 19, it's E, because you see alanine here. The protein sequence is always written from N terminal to N terminus to the C terminus. And number 20, C. So I guess, there's not much of question or doubt here. No questions, right? It's all about, whether you're using charged amino acids or the polar ones, non-polar ones, or so. Question. Yes, those are 20. And I'm sorry, I have to say this, but yes, you're correct that you have to, Yes, you have to. For the structures, it'll be definitely great, if you can memorize all of them. But when you actually think about their structures, all of them are same except for the side chains. And when you look at the side chains, there are some trends there. It's not just the random stuff here and there. They actually recycle, use the similar moiety here and there. So hopefully you can at least distinguish in different amino acids by looking at the structure. And also you can tell that what kind of functional groups it contains and what convert, how those the side chains confer its own property, the amino acid property. I know those are 20, but all right. Number 21. Hopefully it was an easy one too. Number 22. That is correct. What is being racemic? Exactly 50 and 50. Oh, what is question? No, it's the different ones. So non-standard are the basically other amino acids other than essential amino acids or the standard amino acids. And there are two non-standard amino acids that you know, right? Selenocysteine, those kind of stuff, and as well as some other actually that you don't need to concern too much about, as opposed to protein modifications or the amino acid modifications are basically adding some other functional groups onto the standard amino acids. And somebody said, asking about the definition of a racemic. So please go back to the lecture, and it actually says that the definition there. So which is the 50-50 of the different conformational isoforms. Question. About what? Yeah, racemic. So again, you can review the lecture. You're gonna have an idea. Same thing I actually said. 50-50 of the conformational isomers. So. Oh, constitutional isoforms. I'm sorry about that. Yeah, I thought it was kind of some kind of little weird, but number 23. C. And we talked about this, right? Yeah, it definitely, in some cases it could, but mostly not, because it shouldn't affect the protein cores. No, not necessarily. So if protein is denatured, it tends to aggregate and precipitate because it exposes the hydrophobic regions, as opposed to because it precipitates, it does not necessarily mean that they are denatured, because it can still, so it's the soluble proteins are all about the hydrophilic regions exposed on the surface of those proteins. If you actually, mess up with that, then and sort of inhibit the hydrogen bonding with the water molecules, then you can lead to the protein aggregation there too. Is not always what? Exactly, no. Okay, 24. B. 25. What is it? It's D, because I think I heard all different things like A to E. How come? Why is it not E? Gene sequences. Basically, there's a linearity. We talked about this, right? When you have particular DNA sequences, then it leads to the amino acid sequences too, like its own characteristic sequence. So doesn't E the right answer? Isn't E the right answer? No, because? Who said no? I think I heard it. So. That is correct, but we use protein sequence for basically making those like branches, right, in the phylogenetic tree, and it's not the gene sequences, which is DNA sequences, because you can actually get different codons. So there are so many different variabilities there. So you're going to end up having a super like dynamic tree there, as opposed to just to focus on the protein sequences, where it is a little less variable, but still meaningful, right? In the end, you need to consider the protein amino acid sequences and the polypeptides to predict their functionality, their structures, and so on. So we use the protein sequences and evolutionary relationships to get the phylogenetic tree. Number 26, A. I think I've talked about that during my lecture. 27, C, and I'm pretty sure that's straightforward. Good question. So tryptophan is the largest amino acid, but why is it not the right answer? Which is an amino acid. And the reason is because tryptophan has the largest side chain, that's for sure. So it can actually cause some steric clashes and things like that, some conformational limitations and stuff like that in the protein, but it's not necessarily gives the rigidity there, because even if that's a huge side chain, it can still flop here and there, right? As opposed to proline has the direct like covalent linkage to the amino group, so that's why. Yes, like a ring structure there, so that's why. Okay, we're almost there. 28. B, that is a correct answer. Why not A? Phenylalanine. That's nonpolar. Where are those side chains located in the alpha helical structure? Where is it? Outside, inside? Outside. So it really doesn't matter in terms of when you look at the conformation of that. Because even if you have a bulky stuff like a phenyl ring, you can just put them on side. So why not? Any speculations? Because it's bulky. I mean, you're right. Because it's bulky. So it has to have the interaction with the other helix, right? And if the side chains are too bulky, that they can't bring those two helices together really closely. Hard to make the strong interactions between those. So it's hard to hold them as a one big complex. So that's why. 29, A, and 30, D. It's all of them. Pretty much everything can affect the protein structure. So that's it. And if you have any additional questions, just email me or come to the what is it, the office hour. And you can leave if you want. That's totally fine. I'm gonna just quickly go over the points that we discussed. As a review session. So that's why. We talked about the basic structure of the amino acids. Talked about the mirror images, whether it's gonna be form the chiral structure or not, and the exception is glycine. And I'm quickly talking about this, and those might be an important thing actually. Second thing, we talked about the different pKa values, and this is a polyprotic acid, right? The amino acids are polyprotic acid, which means that they have a multiple pK values. We talked about PI values too. And also we talked about how we can get the PI values for the each amino acids based on their classification of their charge. Question. I'm sorry. That's a good question. Is that gonna be on the test? Well, I can't give you the direct answer, but there should be a reason why I'm talking about this. All right, so Protein precipitates when pH equals pi, and that's called isoelectric focusing that we talked about, right? And here's the charge changes. Here's a quick summary, but again, what I want to point out here is you need to identify or distinguish different amino acids by looking at different structures here. That's a good question, actually. So for acidic ones and basic ones, definitely you can apply these equations, right? For rest of them, for example, like serines and threonines and things like that, which can also ionize, since their pKa value is sort of mediocre, it's not going to affect actually too much in terms of getting the pi value. So you can apply plug the original one for those things. All right, again, about the structures and features. Same thing applies to the hydrophilic amino acids, the basic acidic polar ones. And we talked about other special amino acids, prolines and cysteine. These are really important, right? And we talked about the Fischer convention, basically, where you put the molecule in the same way as the L-glyceraldehyde and can tell that whether the hydrogen hydroxyl group is located on the left side or the right side. You can tell it whether it is D form or the L form. And we talked about the standard amino acids, 20 amino acids, including the essential amino acids, and the relationship between the codons and the residues. And here are the essential amino acids, basically. And you should know why they are called as essential amino acids. And we talked quite a lot about the peptide bond formation, where it occurs and how it is formed. What would be the leaving group, which is a water molecule here. And we also talked a lot about the different levels of a protein structure from primary to quaternary structure. Where's the glycine fall? You mean? Oh yeah, it is hydrophobic. Yes. All right, and then here we go. And we talked about the phylogenetic tree, basically, that we use is the protein sequence there, and you can see the evolution, there. And we talked about I don't know if I have here we go, have the orthologs and paralogs and what they mean and how they occur, basically. Different sort of, modes of evolution makes this different types of like homologies. And we talked about the protein sequencing. There are two different methods, right? That we talked about how they actually work, like mass spec and the Edman degradation. So you should, understand how it works. And protein purifications too. And you can use many different properties, like physical properties, chemical properties, like size, charge, binding affinities, and so on, basically. And there were, a bunch of other different kinds of chromatography, techniques that you can use based on the different properties of the proteins. General idea, no, yeah. You don't need to cover the each step, how it works, what kind of chemicals involved, and things like that. You don't need to. So don't worry about it. I mean, a few of you actually, expressed a concern about your grading especially for the first exam, and I'm with you. I understand it, and I want you to succeed, really, for sure. So nothing's gonna be too tricky, too complicated. I'm gonna ask some critical stuff. I'm not gonna just dig up some like textbook like on the corner and just find the little fact there and ask it. I'm not gonna do that, so don't worry about it. And the purification yields, you can use many different methods here, which are all important. SDS page, gel electrophoresis, basically, to separate the DNA fragments based on what? Size. And there's assay-based ones looking at the activities, Western blots, ELISA, ligand binding assays, and so on. And we talked about, again, different types of chromatography, the affinity ones, the size exclusion, ion exchange, and so on. So you should understand how they work. And also talked about the purifying the protein with the tag, like chimera proteins, using the HEAS tag or some other tags. I'm sorry? Oh, yeah, sure. So, I mean, unfortunately, I didn't have enough time to talk about this when I was, explaining this. So I had to do that at the beginning of the next lecture, if you remember that. But basically what it does is that you make a chimera protein with the protein in interest and some kind of epitope tag. And you can use these different kinds of tags like HEAS tag, HA, FLAG, V5, and OLUS, and so on. Those are typically really small epitopes that are purified from the virus, like coding proteins or some kind of receptor proteins and so on, which has a really peculiar shape or the structure there. And you also develop some kind of antibody or some kind of other thing. It can be protein or it can use some kind of property to have the high and specific affinity to those tags to pull down or purify those proteins. So for example, HEAS tag uses this 6 repeats of the histidines, and it has the affinity to the nickel ion. So basically you just coat your resins of your chromatographic column with nickel ions, and then you just let through the proteins, random proteins. You're gonna end up collecting only the proteins that are tagged with the HEAS tag, right? And then you can just elute it with the other like elution reagents like imidazole in this case, which is competing with the histidine because it shares the same structure at the side chain. So once you elute it, you can just use those proteins for further assays or any experiments. Is it clear? All right. So again, we talked about these different structures and their features and so on. So keep that in mind. And we talked about the Ramachandran plots that can basically look at the psi and phi angles and can tell that what type of secondary structures you can get. And that's the plot for showing that the regions where you can form those particular secondary structure. Question. Well, what are we trying to get of this Ramachandran plot? Is that we look at the any kinds of secondary structures, or it can be vice versa actually in terms of looking at it. But looking at the secondary structure, and you can tell that whether this polypeptide making the specific angles there systematically, and also if you measure the secondary, look at the secondary structure, and if you can tell that certain orientation of those amino acids, consecutive amino acids in the polypeptides, you can actually deduce that that will make the certain secondary structure based on this Ramachandran plot. So it shows that. It also shows that the probability of getting beta sheet versus alpha helix, right? Let's say that if a certain protein was located right here, the red dot right here, then it'll have a more probability to become beta sheet rather than alpha helix. So it's an estimation, basically, for that. Can if you can memorize the plot, that would be awesome. I mean, that's like a photographic memory, right? As long as you can understand what it means. All right. And we talked about the secondary structures like alpha helix, beta sheets, and then where they are sitting on the Ramachandran plots and their properties. Yes. That is correct. So not necessarily more stable. It'll be a little bit more like with the lower energy because they have a shorter distance for the hydrogen bonding. So both are pretty much stable, just like the alpha helix. But it's based on the constituents of the polypeptides, basically, and also how the protein is designed through the evolution, too. So it can be either forms, and both of them are stable. Question? All right. And we talked about some higher order structures like the elongated filamentous or fibrous proteins. And we talked about these 3 examples: alpha keratin, collagen, fibroin, and so on, and how they are formed too. We talked about super secondary structure, which are motifs, something like that. They are sort of a functional region. And also the little, upper level would be the domains, right? So we talked about the hydrophilic regions where it applies the hydrophobic core that are inside. So it's going to be the first sort of a formation of the protein. And then talked about the tertiary structure where hydrogen bonding, ion interaction, and all other interactions are important, as well as in this disulfide bonds. And we talked about the different methods to determine the protein structure, like X-ray, crystallography, and the cryo-EM too, which I didn't include here. And we talked about this sort of energy landscape. It's called the folding landscape or folding funnel, where we talked about the entropy changes, internal energy changes, and how you get to the bottom, which is, you can't really see it here, but native states. And we take an example as the GRO-EL, GRO-ES complex from E. coli, which is a chaperone. And that's one example for the quaternary structure or the higher order structure of the protein, where it has a particular function there. And we also talked about its mechanism, how it works, right? And protein misfolding and its relevant diseases too, how it works, prion disease, and so on. And we also talked about the protein functions by taking examples of myoglobins and hemoglobins. There are differences there, basically, but also similarities. So we already talked about them, how they behave, and also how the only hemoglobin has the cooperativity in the oxygen binding, and how it works, sort of conformational changes, allosteric changes, and the differences to the myoglobin too. And we also talked about the other factors that are involved in the affecting the hemoglobin affinity to oxygen molecule, like these. And lastly, we talked about the one example of the disease that is caused by the mutation in the hemoglobin, which is a sickle cell anemia. So these are the mechanisms, what the consequences are. So that's it. Question. Structure of a myoglobin? No, you don't need to. All right, that's it. Good luck with your exam. And there's one thing, some critical thing. is that I included 3 bonus questions. Each of them are 3.33 points, so you can get up to 10 extra points. So if you get all of them correct, then you're gonna get 110 points. Good luck with that. Just a second, I wanted to stop this.